Agriculture has remained the core of human civilization since the beginning. Why? Because of food. All the food chain starts with energy from the sun. This energy is captured by plants. Thus, the living part of a food chain always starts with plants. Plants absorb carbon dioxide from environment and energy from sunlight to form food and oxygen as the most useful byproduct through photosynthesis. Let's dive deeper. Photosynthesis occurs at chloroplast. One of the members of chloroplast, chlorophyll, absorbs sunlight and sends signals to stroma. And here the Calvin cycle begins. The Calvin cycle is further divided into three stages. Stage 1. Carbon fixation. Rubisco 1,5-bisphosphate carboxylase oxygenase is the key enzyme in photosynthesis. 3 molecules of Rubisco enzyme absorb 3 molecules of carbon dioxide to form 6 molecules of 3-phosphoglycerate, and then they are further reduced in stage 2 to form 6 molecules of 1,3-bisphosphoglycerate, and then they form 6 molecules of glyceraldehyde 3-phosphate that are also known as G3B. One of those 6 G3B is used to produce food in form of glucose and other organic compounds, and the remaining 5 G3B are used to regenerate Rubisco enzyme, and here the cycle completes. To synthesize a single glucose molecule, the cycle runs 6 times. But it's not this simple. Do you have any friends who are awesome people, but who also have some kind of bad habit? Maybe they procrastinate a lot, or never remember to brush their teeth. You wouldn't stop being friends with them for these reasons, yet from time to time you might find yourself wishing they would clean up their act. Similarly, during the first stage of Calvin cycle, Rubisco incorporates carbon dioxide into an organic molecule to give energy. But when the light fluctuates, the chlorophyll sends signals to stroma, and then Rubisco creates a major flaw. Instead of using carbon dioxide as a substrate, it picks up O2. Then 2 molecules are produced, a 3-carbon compound phosphoglycerate 3-PGA and 2-carbon compound phosphoglycolate. 3-PGA is normal intermediate of the Calvin cycle, but phosphoglycolate cannot enter the cycle, so its 2 carbon are removed, or better say, stolen from the cycle. To recover some of the lost carbon, plants put phosphoglycolate through a series of reactions. 3/4 of the carbon that enters this pathway as phosphoglycolate is recovered, while 1/4 is lost as carbon dioxide. This process is called as photorespiration. We can clearly state that plants gain energy through photosynthesis and loses energy through photorespiration. Now let's talk about traditional and modern form of farming. Traditional farming is soil-based. The plants take up nutrients through soil, and better to say, they are nature-dependent. The modern one is hydroponics, which is water-based. The roots of the plant are always dipped in water-based solution, and we can change the concentration of this water-based solution. Now let's talk about 2 types of growth in plants, vegetative growth and reproductive growth. In vegetative growth, plant uses nutrients for the development of roots and shoots for a strong plant structure, while in reproductive growth, plant uses nutrients for flowering and fruit formation for the development of marketable crop. Therefore, nutrient concentration is a key factor, since in hydroponics, the nutrients are always available. That's why we are using nutrient concentration as an input for our algorithm.
Now, let's discover how can we use the mechanisms found in nature to produce better quality fruits and vegetables via computational intelligence. Humans always try to find ways to speed processes up and diversify the ways they spend their time. Computers have helped humans speed up calculations and easily analyze data. This thought process led to researchers from Japan publishing a paper in 2003 titled Intelligent Systems for Agriculture in Japan. In the paper, researchers used neural networks and genetic algorithms to optimize the control of a tomato hydroponic farm. Neural networks are a very diverse form of artificial intelligence application used in deep learning and is based on the human brain. As with humans, neural networks are made up of layers of neurons, the amount of which depends on the complexity of the problem at hand. In the most basic sense, neural networks take in data, train themselves to recognize the patterns in the data, and then are used to predict the outputs of a new set of similar data. The magic happens in the hidden layers. Genetic algorithms, meanwhile, coincidentally also derived from our surroundings, have the working mechanism of genetic evolution. The inputs are strings of ones and zeros considered to be the population. The population then undergoes crossover, also known as breeding, and undergoes mutation. That is, DNA codes change based on environmental and behavioral factors. In genetic algorithms, It is simply the flipping of 1 into a 0 and a 0 into a 1. This is exactly what the researchers did. They set up an image processing system in the form of a sensor that would process the images of the total leaf length and the seed diameter to form the total leaf length to seed diameter ratio. Other inputs were light intensity and nutrient concentration values based on traditional farming methods. They used this data to train the neural network to identify how the TLL to SG ratio responded to changes in light intensity levels and nutrient concentration levels. But where was the genetic algorithm applied? A genetic algorithm was actually used to provide the inputs to the neural network. These were the values for nutrient concentration. The neural network was used to evaluate the fitness of these values and used to predict the behavior of the TLL to SG ratio based on any combination of 4 step points of nutrient concentration. Traditionally, for a 4-step growth process, the nutrient concentration is increased for every step. However, according to the optimized values that were given by the system of genetic algorithm and neural network, interesting things were observed. At the initial stage, a moderate value of 1.4 was selected, followed by a remarkably lower value of only 0.3. The third stage spiked up to 1.6 and the last stage peaked at 2. Now, let's figure out why did the computer think this was a good way of going about things. Actually, the higher value of nutrient concentration in the first step encouraged vegetative growth initially, while the far smaller value in the second stage was effective in suppressing excessive vegetative growth during the seedling stage. This is of course because traditionally in hydroponics, the roots are always in a suitable environment for the uptake of nutrients. The third and fourth steps help accelerate reproductive growth and give the fruit of our efforts.